Good morning, class. Hopefully you guys are doing okay with studying for this week. Today we're going to continue immunology, specifically the adaptive immune system. This one's also going to be a little bit of a dense lecture, but I do what I always do is layer it on so it's simple to understand. At any point, if you guys have any questions or clarification, ask in the chat or unmute yourself. The whole point of me going over all of this immunologies is I really want you guys to have an understanding on how your body is able to fight off infections. But more importantly, this is going to be a prelude for you guys to be able to understand how some of the therapies we have against COVID are going to work, specifically some of the different kinds of vaccines that are out there. So please make sure you guys have a clear understanding of both the innate and the adaptive, but more the adaptive because the vaccines are going to be more implicated in trying to develop this memory set of adaptive immune cells. So for today's outline, I'm going to go ahead and give you a brief overview of what lymphocytes are. And then we'll go ahead and talk about some of the unique properties that the adaptive immune system has. And then I'm going to go ahead and layer this on by giving you an overview of T and B cells and how they're able to quote unquote see antigen, because it's quite different than what we discussed yesterday when it came to innate immunity. And I'm going to also talk about what happens when you get a secondary infection. This is going to be important to understand because it's going to go hand in hand when we talk about vaccine therapy next week. And then I'm going to go ahead and talk about how our immune system responds to COVID and how some patients end up succumbing to the infection. What I really want you guys to understand, so it's going to be a lot of information. And again, I'm going to layer it on over and over again. You're going to hear me repeat a lot of stuff, and hopefully at some point, some of it sticks. But what I want you to understand are what is the differences between B and T cells. Yes, they're both part of the adaptive immune system, but they have two different distinct functions. I want you to understand how they're able to recognize antigen, how they're able to see that a pathogen is present, and understanding how the B and T cell receptors are implicated in this and how the MHC complex or major histocompatibility complex is related to T cell receptor interaction. I want you to understand the structure of an antibody and the important parts it plays when it comes to trying to fight a foreign invader. And then understand what happens to the immune system when a secondary infection is present. And then finally, I really want you to understand why some COVID patients succumb to infection. Again, if you understand the learning objectives, then you're going to do fine on the exam. So yesterday I focused mainly on the innate immune system, so the myeloid origin cells. So for today, I'm going to be focused solely on T and B cells, so the lymphoid origin cells, and basically understanding their function and how their function is connected to innate immunity. So lymphocytes come from a common lymphoid progenitor cell. So, they branch off in the bone marrow by this particular stem cell. And the stem cell can actually differentiate into three different cells. So B cells, T cells, and natural killer cells. I'm not going to talk about natural killer cells in this class. I think it's beyond the scope of the understanding. So don't even worry about that. But these three cells are they're all considered lymphocytes because again, they're from the lymphoid origin cells, but it's only the B cells and the T cells that are considered adaptive. Natural killer cells are not adaptive immune cells. They function more as innate immune cells. It's important to note that B cells produce antibody and that they only produce it once they differentiate into this specific subtype of B cells called plasma cells. As opposed to T cells, I'm going to talk about that there's two different kinds later in the lecture. Basically, one type is going to mediate killing of infected cells, while the other set of T cells are going to play a role in increasing the activity of both innate and adaptive immune cells. And so this is going to help, educate the innate immune system as well as the adaptive, but innate immune system to help recognize pathogens more efficiently. Every T and B cell has a unique receptor that only recognizes one type of antigen. So basically, it's thought that for every kind of pathogen that exists, you have one kind of T cell that is able to recognize that pathogen. And I'll go more into that when we're actually talking about some of the receptors that are implicated in that. So yesterday, you guys saw this slide. So just, a quick reiteration of, how the innate and the adaptive kind of connect. So, you have, we'll use bacteria infection in this particular case. So you get a cut on yourself. There's an introduction of bacteria into the wound. You have immune cells that are already living within the actual skin itself. So they're going to be the first responders to basically try to clear up the infection. Dendritic cells in particular are going to be able to phagocytose or eat some of this bacteria and take it to a secondary lymphoid organ. So the main one you're going to see in this lecture are lymph nodes. So secondary lymphoid organs are where the majority of your adaptive immune cells live, so your B and T cells. And the whole point of them living here is so innate immune cells from the periphery or outside of the secondary lymphoid organs can run to the secondary lymphoid organs, such as the lymph node, and present antigen to your B and T cells. Once your B and T cells are educated, then they can go ahead and leave the secondary lymphoid organ, so here the lymph node, and go to the site of infection so it can do its job. So for the duration of this lecture, we're going to be focused on what's happening in the second part of the infection. So just to emphasize, secondary lymphoid organs is where your adaptive immune cells live. Once they interact with antigen, then they can go ahead and leave their secondary lymphoid organ so they can go ahead and do their job. And so remember, distinguish this between primary and secondary lymphoid organs. Primary, again, is where, highlighted in yellow here, is where your immune cells are going to develop. Once they're already developed, then they're going to go to some of these locations so they can basically be on standby for a pathogen that may come into contact with them. So I left off on this slide yesterday talking about this is where adaptive immunity begins. So after an innate immune cell ends up coming into a secondary lymphoid organ, such as the lymph node as depicted here, it's going to interact with both T cells and B cells. So for the first part, we're going to be focused mainly on the T cells, and then we'll kind of connect all of that. So this is basically a zoom in of this. And so when innate immune cells are interacting with T cells, there's a couple of things that need to happen in order for the T cell to actually get activated. For this lecture, I'm only going to be focusing on signal 1. Don't worry about learning signal 2 and 3. That's beyond the scope of the class. I think you would have to have a more understanding of some of the cytokines that are present. What I want you to understand and focus on is how our T cells are able to recognize antigen. So once an activated innate immune cell arrives to the lymph node or other secondary lymphoid organs, it's going to present it to a naive T cell. And a naive T cell is just a type of T cell that has never seen antigen before. So it's basically just waiting for it to come in contact with a antigen presenting cell. And most of them are going to be innate immune cells, but we'll get more into that in a second. So in order for the T cell to recognize antigen, it needs its T cell receptor. So here I have highlighted in yellow what the T cell receptor looks like. And so the T cell receptor is only going to bind to this thing called major histocompatibility complex. And there's 2 of them, which, I'll distinguish them in a second. But for understanding right now, just understand the TCR on a T cell receptor binds to an MHC complex. And on this MHC, it's going to be loaded with antigen. So again, antigen is just a little fragment of the pathogen that is invading. And so this process needs to occur in order for the T cells to be able to actually know that an infection is happening. Unlike going back to the innate immunity where they have these pathogen recognition receptors, where they can basically just pick up random things from the environment, T cells do not have those. They need this interaction in order to actually see that a pathogen is present. So, going a little bit more into what an antigen is. So I kind of gave you a preface of that yesterday. So the immune system is going to recognize this as foreign or non-self. And generally, these are going to be a short peptide sequence. So, this is, if we go back to lecture 3, I think it was, peptide chains are just a sequence of amino acids that are stuck together. And both B and T cells recognize this. However, it's only B cells that can recognize carbohydrates, so sugars, and nucleic acid sequences. So carbohydrates are typically going to be found on the surface of bacteria, and nucleic acids are going to be found in viral cells and bacteria if they end up lysing out. But usually B cells will end up interacting with viruses if their nucleic acids are somehow in the extracellular environment. It's only a portion of the antigen, though, that's recognized by the T and B cells. And so here I haven't read what an antigen looks like. So it's, simplified. It's just, again, a piece of the overall pathogen that's present. So if it's a virus, it's a piece of the virus. It's a bacteria, it's a piece of the bacteria. But the actual part that the receptor binds to, and see here I have a B cell receptor. It's the actual name is called an epitope. So the epitope is the part of the antigen that the B or T cell receptor is able to recognize from the antigen. So for B cells, antigen can be picked up from the extracellular environment. So if there's free-floating bacteria particles or whole bacteria, or if there's viral particles or whole viral particles, it can actually bind to it via its B cell receptor, so this antibody, and it can basically eat the pathogen, so phagocytose. Once it ends up eating, if it is indeed a whole pathogen, it's going to end up eating it, phagocytosis, and then there's going to be lysosomes within its body that are going to chop it up into pieces or fragment it, and then a piece of it is going to be presented to the T cell as antigen. Whereas the T cells, they need, again, this T cell receptor, and they can only recognize or see the antigen when an antigen is loaded onto that major histocompatibility complex. And these MHCs are expressed by antigen presenting cells. So if a cell is presenting antigen to a T cell, it is called an antigen presenting cell. So there are two different types of major histocompatibility complexes. So there's MHC1 and MHC2. So for MHC1, basically this is expressed on all cells in your body that have a nucleus. And so all of your cells in your body have a nucleus except red blood cells, and it has to do with the whole differentiation process. But the whole point of this is, the reason why all cells have an MHC1 is because all cells can get infected, theoretically. And so if a cell is infected, it wants to communicate to the immune system that I'm infected, kill me, basically. And so they do this by taking some of the antigen that's within them, or the viral particle, or whatever is the intracellular organism. They take a piece of it, put it onto the MHC, just as you see here in green, and then put it on its cell surface so a T cell can come and recognize it. And then once the T cell comes and recognizes it, it basically can kill it. And so we'll get more into the kinds of T cells that are specific for doing that. Whereas the MHC class 2 molecules, these are expressed by only 3 cell types: macrophages, dendritic cells, and B cells. And these are known as professional antigen presenting cells. And the reason why they're considered professional antigen presenting cells is because the 3 of these cells have phagocytic capabilities. They can eat pathogens whole and then process them so they can basically put the antigen onto their MHC2. And with MHC2, if a cell is putting an antigen on their MHC2, they're not telling the T cell to kill it. They're telling the T cell, here, there's antigen present, there's a pathogen present, go ahead and get activated. And so that's why they're called professional, because these are the ones that don't actually get killed. They're not telling the T cell to kill me. They're just telling the T cell there is a pathogen present. Whereas with the MHC2, they're telling a certain kind of T cell, I'm infected, kill me, so this pathogen doesn't have a place to live. So here are some of the professional antigen presenting cells highlighted. So again, there's three main ones: dendritic cells, macrophages, and B cells. And these are the ones that all express this MHC class 2 molecule. It is important to note, because they all have a nucleus, they also express MHC1. So if, say, they were infected themselves, they would be able to tell a specific subset of T cells that I'm infected, kill me. Versus if they're not infected and they're just uptaking antigen from the environment, then they can go and tell the other subset of T cells, hey, there's pathogen present, get activated, and start alerting everybody. And so the type of T cell in particular that these professional antigen presenting cells talk to are the CD4 helper T cells. So of course, I'm not going to have you, memorize this whole pathway. That's ridiculous. But what I want you to understand, or the reason why I'm showing you guys this, is to understand the two different ways these MHCs are put on the cell surface of a cell. And so with MHC class 2, which I just talked about, where it's just a dendritic cell, a macrophage, and B cell, they're able to take extracellular antigen, so antigen that's in the environment. And so this can be either fragments of the pathogen or whole pathogen. And they basically endocytose it or phagocytose it and then fragment it. And then when they fragment it, they load it onto the MHC2 so they can talk to the CD4 helper T cells. As opposed to the MHC class 1 molecules, basically there's an intracellular pathogen that ends up getting into the cell that ends up infecting it. And then we have specific processes in our body that are able to fragment it itself. But this happens when the cell is actually being infected. And so because some of these pathogens hijack some of our organelles in our replication machinery, in the process of all of this occurring, some of the antigens, so viral or bacterial or whatever it is, will end up getting loaded onto an MHC class 1 instead, and then putting it onto its cell surface. And this will end up telling everybody that I'm infected, have the CD8 T cells come and kill me. Someone asked, is there such thing where infected cells can be cured? That's a good question. Not to my knowledge. I would say the only way that it is possible, and I guess this is my knowledge in terms of parasitic infection, is after the adaptive immune system, or let me back up. If, say, an innate immune cell is infected with a parasite or something like that, certain parasites, if they're intracellular, they form basically these vacuoles or these membranes that are like their homes that protect it from different enzymes that'll end up killing it. And so what ends up happening is once the adaptive immune system ends up getting educated, it's able to secrete certain cytokines that will end up telling the cell, basically make certain proteins, because the whole signaling process ends up occurring, and it ends up destroying the home of the parasite. And so if the home is destroyed, then certain enzymes within the cell can actually kill it. So that would be the only way that I can see it, an infected cell being quote unquote cured. But most of the time they're actually killed just to prevent any spread of the disease. So if a cell that doesn't replicate often get infected, it could be lethal. Yeah, for sure. And so this happens a lot when it comes to like neurons. Neurons are a cell type that don't replicate very often. And so if they end up getting infected, say like you have some kind of brain infection with a parasite or a virus, sometimes these T cells, once they're recruited to the brain, will end up killing some of these neurons. And I think it was recently discovered in like within the last 10 years that we actually have stem cells in our brain, so we can regenerate some of these, but it's a very slow process. And so that's why some people that get brain infection, they end up succumbing to the infection because of the amount of damage that's done. All right, so we'll go ahead and emphasize this point one more time. So the MHC needs to be loaded with antigen so the T cell receptor can bind. So again, the T cell receptor can, or the T cells can only see antigen with their T cell receptor. So here we have in blue a T cell receptor, and then in these respective different colors, yellow and green, we have a specific MHC complex, and these are going to be found on antigen presenting cells. Like I was saying at the beginning of the class, every T cell receptor is different. And so because it's different, think of it as a puzzle. It's only going to be able to fit a certain piece in there. If it doesn't fit a certain piece, then the T cell isn't going to recognize the pathogen and it's not going to get activated. And so here we have a pathogen that actually fits perfectly with this particular T cell receptor. So then downstream function of the T cell actually getting activated can happen. But if the binding pocket doesn't fit perfectly like a key, then none of that activation is going to end up happening. And then it's also important to stress this. So if a T cell is present around like a bunch of bacteria or viruses, but there are no antigen presenting cells, the T cell will not know that an infection is occurring. This process needs to happen in order for the T cell to know that there's infection present. So I'm going to emphasize two different types of T cells. So in reality, I'm an immunologist by training, so there's actually shit tons of T cells that exist. But for simplicity, the two that you need to know are CD4 and CD8 T cells. So we're going to start with CD8 T cells as they're relatively simple. Basically, the other name for them that people will use interchangeably is cytotoxic T cells. And so these particular T cells are only going to recognize the epitopes or that part of the antigen that is loaded onto an MHC1. So MHC1 will interact with the T cell receptor of CD8 T cells. And so the whole point of this CD8 expression is it functions as a co-receptor, and it basically stabilizes the interaction between the MHC1 and the T cell receptor. And so we can see this here. We see an antigen presenting cell that has MHC class 1 loaded with antigen highlighted in red, and then its T cell receptor is recognizing this, but this CD8 T cell needs to have this co-receptor in order to stabilize this interaction. If it doesn't have this CD8 present, then this binding does not occur. Once a CD8 T cell ends up recognizing the right antigen that's being expressed by this antigen presenting cell, it's going to facilitate killing, because as I mentioned before, MHC1, once it's loaded with antigen, it's telling the CD8 T cell that I'm infected, kill me, so there isn't any more replication that can occur. And so here we have a viral infected cell, making more viral particles, and then basically the cytotoxic T cell is going to end up performing this interaction, and then it's going to end up killing the infected cell. And so if you kill the infected cell, then the virus or whatever it is no longer has a home to replicate. So how do CD8 T cells actually kill these infected cells? So as I already pointed out, they need to recognize the right antigen on the MHC1, and you have to have this co-receptor being able to recognize that. And once this interaction happens, the T cells are going to end up basically secreting a bunch of cytotoxic molecules, so stuff that's toxic to cells. So some examples of these are perforins and granzymes. And basically, these are going to end up poking holes in the membrane, as well as basically once the cell membrane is compromised, then the cell is going to be programmed for cell death or apoptosis. And the game that I have you guys playing today for your worksheet will emphasize this quite a bit when it comes to you trying to kill infected cells. So hopefully, that'll help further help you understand these concepts. So the other main the other main T cell are CD4 T cells. And these are known as the helper T cells. So these aren't going to kill any cell. What they want to do is they want to recognize antigen, as highlighted here, that is being loaded onto an MHC2. And again, the only cell types that express MHC2 are macrophages, dendritic cells, and B cells. And so the whole point of this is so the CD4 helper T cells can interact with those 3 professional antigen presenting cells and basically direct them on what to do. And so here we have an example of a helper T cell interacting with a macrophage. And so once this happens, if say the bacteria is indeed infected with bacteria, it can upregulate some molecules after the signaling ends up occurring and actually facilitating some of the clearing of the pathogen. So as someone was asking before, it's a way that some of this curing can happen. But again, the majority of this is driven by CD8 T cells, which will kill. And then what CD4 helper T cells will also do is activate B cells. And so before I get into the actual B cell stuff, just to reiterate some of these points, MHC class 1 is expressed by any nucleated cell. So again, that's all cells in the body except for red blood cells, which lack a nucleus. These antigen presenting cells, which express MHC class 1, will present only to CD8 T cells. And so we can have this example here. CD8 T cells recognizing the MHC class 1 with the CD8 co-receptor stabilizing this interaction. And again, this whole interaction is communicating with the T cell that the antigen presenting cell is infected, kill me. As opposed to the MHC class 2, which is only expressed on these 3 cell types, which are called professional antigen presenting cells. And they present antigen to only CD4 T cells. And that's to help facilitate the further driving of the immune system to clear the pathogen. And just like CD8, CD4 is also a co-receptor that helps stabilize the T cell receptor MHC class 2 interaction when this antigen presentation is occurring. If you need help remembering, which T cell binds to which MHC class, this is a mnemonic that I find very helpful. CD8 T cells will bind to MHC class 1 and 2 and CD8 4. So 1 times 8 equals 2 times 4. So never forget it. So what ends up happening after this interaction actually happens? So there's a couple of other signals that the T cell needs to get activated, which I'm not going to quiz you on or get into depth because I feel like it's beyond the scope of this class. But what you need to understand is once this interaction begins, the T cell is going to undergo something called clonal expansion. And clonal expansion is just that one T cell making many copies of itself. So like I said, every T cell receptor is very unique to the kind of pathogen that it recognizes. And so because there's only one type of those, once this antigen presentation occurs, that one T cell that has that one receptor that recognizes that one specific protein or antigen is going to multiply itself. And after it ends up multiplying itself, then it can go ahead and leave the secondary lymphoid organs, such as the lymph nodes, and basically go and hunt down the pathogen of interest. So this is highlighting CD8 T cells, and they like to kill. So they're going to go to the site of infection and kill off any cells that are infected to prevent further replication of whatever pathogen is present. It's also important to understand that the T cells aren't just killing, they're also going to be telling, and this is the CD4s, are going to tell the innate immune cell, get better at eating the pathogen. And I'll go ahead and talk about that in a second when we're covering B cells. Once the pathogen is cleared, instead of clonal expansion happening, there's this term called clonal deletion, and I'm not going to quiz you on any of this, but what I want you to understand is you don't want all of these T cells that have clonally expanded still to be around once the pathogen is cleared. So a good portion of these, I think it's up to like 90%, end up undergoing apoptosis, so programmed cell death. But a small fraction of them are going to end up surviving. And this small fraction of educated T cells are going to be your memory T cells. And so these memory T cells are going to be important when it comes to seeing the pathogen again. And I'm not going to quiz you on the different kinds of memory T cells that are present, but there are 3 different types that are present, if in case you wanted to know more about them. And if you want to know more about them, feel free to come to office hours because T cells are my specialty. Yeah, so that's that. I think I had a question. Does one T cell have multiple receptors that bind to different antigens, or do they just bind to one specific? Good question. They only bind to one. So though they have like, I forget, like maybe 10,000 copies of T cell receptors on their cell surface, that T cell receptor is all the same. So they can only recognize one specific antigen. All right. So now let's go ahead and talk about B cells. So B cells are going to be what become plasma cells that secrete antibody. And so B cells are going to be the other important adaptive immune cell that is needed when it comes to fighting against foreign invaders. And so, like I mentioned before, it's a professional antigen presenting cell, and all professional antigen presenting cells have the capability to phagocytose pathogens. And so it can be a whole pathogen, as you can see here. So, you know, this is just we'll say a viral protein or a viral particle. Or it can just, if there's fragments of it in the environment, it can recognize it. Once it recognizes it, it can go ahead and internalize it or phagocytose, and then fragment it, and then basically load it onto its MHC2. The MHC2, again, is going to interact with the T cell receptor on CD4 T cells. Once this interaction occurs, it's going to communicate with the B cell to do 2 main things. One, just like the T cells, it's going to cause it to clonally expand because, again, you know, the T cell receptor is TCR, and the B cell receptor are these antibodies. And basically, these antibodies are just like the T cells, only recognize one specific thing. And so because its B cell receptor only recognizes one specific thing, it needs to make many copies of itself. So it's going to clonally expand. And a fraction of those cells that are clonally expanded are going to end up differentiating or turning into an antibody secreting immune cell called the plasma cell. So plasma cell is just a specialized immune cell or B cell that is able to secrete its antibody. So here I have an antibody structure. And so these are things that I do want you to understand because it's going to be important when it comes to understanding how some of these COVID therapies work when I talk about them next week. So antibodies are just B cell receptors that are in the secreted form. So a B cell that's not differentiated is going to have its antibody intact. It's going to be on its surface. Whereas a plasma cell is going to end up secreting its B cell receptor. And that's all it does. It just secretes a lot of antibodies into the blood so it can go ahead and bind to antigen. All antibodies have this general Y structure, so it's important to understand that. And there's 2 main parts to this antibody that I really want you guys to know and understand because they both have very important functions. So in the variable region of the antibody, this is where your receptor is actually going to recognize the antigen. And like I said, every B cell is very specific, or every B cell receptor is very specific. And so every B cell is going to have a different variable region, so it can recognize the specific antigen of interest. So make sure you understand this variable region and what it does. This is what actually binds to the pathogen or the antigen. Whereas the other part of the antibody is known as the constant region. And within some of this constant region is this FC portion. And the constant region does not interact with the pathogen itself. That's only the variable region. What the point of the FC portion of the constant region is to help immune cells, specifically innate immune cells, recognize that that there's pathogen present via its FC receptors, and so it can actually phagocytose or eat anything that has antibody bound to it. So you have, macrophages are a big thing, but most innate immune cells have these FC receptors, and they're just circulating within your blood, trying to find anything that has antibody attached to it. And if it has antibody attached to it, it's going to be usually some kind of pathogen. And so the macrophage is going to utilize its FC receptor to bind to the FC portion of the antibody, so it can end up internalizing it. I hope that made sense. Is there a question? Are antibodies the MHC2 of B cells? No. So antibodies are, if you're comparing if you're making it analogous to something, antibodies are going to be analogous to the T cell receptors. So B cell receptors and T cell receptors are the things that recognize antigen. The MHC2 is how the T cells are going to interact with antigen presenting cells, specifically antigen presenting cells. Does that make sense? I hope that makes sense. And if not, feel free to see me after class and we can go ahead and talk in greater depth. And so like I emphasized earlier, when antibodies are binding to antigen, they're going to bind only to the Y portion of the antibody. And unlike T cells that only recognize specific protein sequences or peptides or epitopes or antigens, they're all, used interchangeably. The kinds of antigens that B cells can recognize is not only proteins, but also sugars and nucleic acids. And so these are typically going to be things that are found in the extracellular environment. And so like I mentioned earlier, a lot of bacteria have sugar residues on their cell surface. And so if this antibody recognizes some of this bacterial sugar, it'll be able to stick to it and then eat it. And then the same thing with nucleic acids. So say there's a bacteria that ended up lysing and some of its genetic material is out, or same thing with the virus. If any of that nucleic acid is free floating within the body, your antibodies will be able to attach to it. And it's crazy because antibodies can basically recognize 10 to the 11 different kinds of antigens. It's crazy that it's able to do this. And there's a whole, somatic recombination stuff that ends up occurring. We're not going to get into that. If you want to know more about it, come to the office hours. But yeah, even though every B cell can only recognize one pathogen, it can undergo some kinds of mutations within the actual antibody structure where it can recognize different molecules. Is there a danger of antibodies binding to cells that are not infected? Good question. Yes, there is. And so that's where autoimmunity comes into play. And so usually there's a whole selection process that happens within the bone marrow for B cells, and the same thing with T cells in the thymus, because you don't want their receptors recognizing ourselves. And so sometimes, and a lot of scientists still don't know why this is, but sometimes these receptors, so the antibody or the B cell receptor or the T cell receptor, will have a high affinity for our own self. And that isn't good, because if, say, a CD8 T cell is recognizing it's recognizing self, it's going to end up killing that cell. And then the same thing with antibodies. Instead of actually killing the cells, it can do a couple of things. One, the antibody, if it's bound to a free-floating cell, then macrophages can come in and eat it. You don't want that. Or it actually can bind to certain receptors and activate those receptors. And so these are all well-studied things in autoimmunity diseases. And if you want to know more, come to office hours, but I don't want to, confuse people with that. But autoimmunity is just our own immune system attacking ourselves. And I'll end up talking a little bit about autoimmunity when I'm talking about diabetes, as there's type 1 and type 2, and type 2 is an type 1 is an autoimmune disease. And so I'll talk about, self or our immune system recognizing ourselves that can lead to disease. So here I have a list of the different kinds of antibodies that exist. So there are 5 different types that are found free floating in the blood. So that's all, being secreted by the specialized B cells called plasma cells. And so I guess I can walk you through some of these. The 2 that I really want you to focus on are Igm and Igg, because I'm going to end up re-emphasizing some of this when we're talking about detecting COVID next week, and having an understanding of these antibodies and what they mean will help you understand how we're able to detect this, and not just with COVID, with other pathogens. So I don't know if anyone in the class has ever gotten their antibody titers titrated for a specific pathogen, but typically doctors will look at Igm and Igg, and I'll go ahead and distinguish the differences in the, I think, the next slide. So with Igm, once an infection first occurs, your B cells are going to start differentiating into plasma cells, and then they're going to start secreting Igm first. And so Igm, notice how it forms this pentamer, this 5-pointed structure of antibodies. And the reason why it does this is because of the variable region of the antibody. So the variable region, is going to bind to pathogens, but there's something called specificity and what's the other word? Not avidity, affinity. Affinity for a pathogen. So affinity, think of it as a magnet. And so if this has low magnetism, then it's not going to be able to bind very well. And Igm doesn't bind to pathogens very well. It can recognize it, but it doesn't bind to it very well. And so that's why there's 5 of them. And so once there's some, it's called isotype class switching, where there's a recombination of the proteins within the actual antibody, it'll end up switching to this other type called Igg. And Igg has a high affinity or a lot of magnetism to the pathogen of interest. And so that's why there's only 1 of it by itself. And so these 2 are going to be very important to understand when it comes to fighting against infection, but also understanding how we're able to detect if a disease is ongoing or if you previously had it. IgE, I mentioned yesterday when we were talking about mast cells, and this is going to play a role in allergic reactions. There isn't too much known about IgD, so I'm not going to end up, asking you anything on that. Just know that IgE is associated with mast cells. And IgA is specifically secreted mainly in the intestine or anywhere where there's a mucus. And notice how this particular antibody forms a dimer, or 2 antibodies stuck together. And it's similar to the the concept of affinity or its attraction to a specific pathogen. But most of the time, these are just put into your mucous membranes, and we have tons of that in our intestine, and that's to really help fight against different kinds of pathogens that are able to make it to the intestine. It's also important to know that IgA is secreted in breast milk, and so it's one reason why you really should have your baby breastfed, because this is a way that babies can develop immunity early on. It can because it's found in the breast milk, the baby can actually eat it and get incorporated into the mucosal area of its intestines. So IgM versus IgG. And so, as I mentioned, it's the first antibody that is made to fight against pathogens. And because it has low affinity for pathogens or low magnetism, it's going to form that 5 point structure, pentamer. As the B cells become more educated, they're going to undergo that thing that I talked about, class switching. I'm not going to quiz you on any of it, but it's just the name that is for undergoing the rearrangement of the proteins within the actual antibody structure. And when this happens, it basically will switch to the IgG form of the antibody. And so the IgG is going to be what's more specific for the pathogen. And so here I have a nice graph kind of depicting on when you have certain levels of IgG, or respective antibodies. So say you have a viral infection that occurs, and then, the immune system is going to end up getting activated, and then your adaptive immunity eventually starts to take over. First off, the IgM is going to end up getting produced. And then once that class switching begins, then your IgG ends up getting secreted. And so I'll emphasize this again next week. But when doctors want to see if you say, this is a perfect way of testing for COVID, if you've had it or have ongoing infection, what they do is they look at these 2 particular antibodies within the blood. And so if they check your blood and you have IgM, that means that you have ongoing infection. And if you only have IgM, then that means, it's in the early stages or the acute stage of infection. Now, if they test your blood and you don't have any IgM and you only have IgG, that indicates that the infection has been cleared, but you have had the infection before. Now, if they measure your blood and you have both of these in here, you basically have, you're in the process of clearing the infection, or you've already cleared it, but there's still, residual IgM still left over. So usually somewhere in this part of the infection stage, if there's clearance, you'll have both. And then with IgG, you basically have it for life, or it really depends on the pathogen. We don't really know with COVID or, the vaccine. Currently, I think right now it's estimated to up to 6 months. I'm sure it's longer. A lot of these antibodies last months to years in your blood, and that's going to help facilitate with a memory response. Let's see, question. Is this specific for COVID, or can this apply to other viruses? This can apply to any other pathogen. So it's all dependent on what antibody the doctor is looking for. So say if you're interested in seeing if you've been infected with a parasite, then they'll look specifically for those IgMs or IgGs for that specific parasite. And then from that, they can go ahead, depending on which one you have present, they can determine if you have an ongoing infection, an infection that's about to be cleared, or an infection that you've already had that's been cleared. But just to relate this to COVID, that's why I ended up kind of just keeping it simple, but this can be any pathogen really. But it is important to note that it's very expensive for some of these. And I know a lot of insurances don't cover this, so a lot of people don't actually get tested for that. When it comes to antibody defense, the whole point of an antibody is to help clear the infection. And it does this in 3 different ways, but I'm not going to go into the complement activations beyond the scope of this class. And there's a lot more that you would need to know to really understand this. But for simplicity, neutralization and opsinization are 2, processes that I feel like you guys should understand. And they're just words, understand what they mean. So with neutralization, just how we have here, basically what the antibody's purpose is to prevent the pathogen from being able to bind to your cells, or to get into cells, or to mediate some kind of response. So for example, when it comes to viral infections, we've all know at this point that viral infections need some kind of receptor or receptors to get into a host cell. If a virus is surrounded by antibody, then it's going to be neutralized, because all of that antibody surrounding that viral particle is going to inhibit the virus from actually being able to bind to those receptors and facilitate entry. So that's what neutralization means. So it just means it prevents the pathogen of interest from causing harm to your cell. In this particular diagram, they have an example of bacteria toxins. So bacteria are able to secrete specific toxins that can cause disease. And so in order to prevent those toxins from affecting certain receptors on a cell surface, antibody can go ahead and bind to those toxins and prevent them from actually binding to the receptor of interest and preventing disease that way. The other main function that antibodies use to fight against pathogen is this fancy word called opsinization. And I had an immunology professor once describe opsinization as like adding salt and pepper to a pathogen so it's easier for innate immune cells to come and eat it. It makes it tastier. And so, like I was saying before, innate immune cells have a certain receptor called an FC receptor. And the FC receptor is going to bind to this FC portion of the antibody. And so if there's antibody that's surrounding this pathogen, then it can go ahead and bind to the FC portion of the antibody and facilitate phagocytosis. And because this antibody is surrounding the whole pathogen, the the innate immune cell will be exempt from being infected. And so once this whole process ends up getting phagocytosed, it ends up basically using some enzymes to fragment it. And again, do not worry about this complement. Very complicated and beyond the scope of this class. So what happens when we actually encounter the same pathogen again? So basically, once we see, once we have a primary infection or we get infected once, as I had mentioned before with the B and T cells, you develop memory cells from those. And so after you develop the memory cells, a lot of them are still going to be in the secondary lymphoid organs, but some of them are also going to be patrolling the blood, looking specifically for the pathogen. And so if you were to get a secondary infection or a second infection, what ends up happening is once these memory cells end up recognizing that there's a secondary infection, they're going to be able to not only respond faster, but the amount of response is going to be a lot stronger. And so this happens for two reasons. The fast component has to do with, remember how I mentioned in yesterday's class, it takes about 5 days before your adaptive immune system starts to actually kick in. And then after it starts to kick in, it takes some time for the whole clonal expansion to begin. When you already have educated memory cells, that whole innate to adaptive doesn't need to happen. It can just automatically get stimulated and then start to clonally expand. So there's a bunch of these memory cells that come and kill off whatever the pathogen is. And then because it's not only faster, when you're developing all of these clonally expand memory cells, their receptors have a stronger affinity or a stronger magnet to whatever the pathogen of interest is. And so there's a correlation between how strong a T cell or B cell receptor binds to a pathogen and the amount of cytokines or effector molecules the adaptive immune cells are able to produce. And so here we have this nice graph showing a concentration of antibody at the beginning. When you first get exposed to a pathogen, you don't really have anything. And then eventually, it peaks and then you start to level off some of your antibody. But if you end up getting re-exposed, a lot of that antibody will end up going back a lot sooner and a lot faster than the primary infection. And so this is important to understand because this is basically how our vaccines are designed, is instead of actually having a primary infection with COVID, we're educating our adaptive immune cells, so being T cells, to recognize viral proteins as if we had a primary infection. And so when we experience a secondary infection, or in this case, if we've got vaccinated and never been infected, it'll be a primary infection. But because we have these memory cells already present, this secondary response can occur because you already have those memory cells generated. And I'll end up re-emphasizing this next week when we're talking about vaccines and how this is connected to the adaptive immune system. But this is just a preface to that. So what happens when your immune system goes into overdrive? And so this is in the context of not autoimmunity, but just a normal infection that your body's trying to clear. Somebody asked, for me, the COVID vaccine was the first one I had with 2 doses. Why did we not do that for other vaccines? So I'll emphasize this a lot more next week. It has to do with the technology that's used. So a lot of previous vaccines that we've used in the past they're called heat inactivated, or they're weakened viruses. And that's similar to some other COVID vaccines that are on the market. And so usually once you have a weakened version, then you can mount a response against that. With the Pfizer and Moderna vaccines, they're mRNA vaccines. So they're going to function a little bit differently, and I will get more into depth on how that works next week. But basically, because it takes more time for some of these viral proteins to get recognized by your immune system, you want to stimulate twice. And there are some other vaccines where you have booster shots, and that's to just make sure that you have that memory response or memory cells still present, because after some time, some of the memory cells do start to die. And so if you end up stimulating them with particular antigen, then you'll be able to cause them to grow again, and then keep a maintenance of them. So when those cells become educated via vaccine, do they kill infected cells faster? Yes. So when the vaccine educates your your adaptive immune system, once you get the infection, it's going to be able to kill it a lot faster. And that's why people should be getting vaccinated, because you're not going to have that 5-day delay of virus basically replicating in you and then mounting a response, versus you already have the memory response, so they can go out and find the virus a lot faster and basically kill any cells that are present, or if there's cells that are infected, kill those cells. All right. So when the immune system goes in overdrive, when it's trying to clear an infection, there's this thing called cytokine storm. And so this happens when there's an overproduction of cytokines and chemokines. And as we'll remember from yesterday, and really understand what these terms mean, the cytokines and chemokines, they have specific functions. The chemokines are going to recruit immune cells to the site of infection, and the cytokines are going to facilitate entry of some of those immune cells, as well as killing of infected cells. And so with some COVID patients, you basically have a lot of this going on. You have the virus getting into your airways. Here we have sections of your alveoli of your lungs, which expand when you breathe. And so once the infection ends up getting into your lungs, your immune cells are going to come in and start producing these inflammatory signals. Well, when too many are present, and this a lot of the time happens if there's a lot of virus that's within your lungs, you're going to have this positive feedback loop where you're basically telling, hey, come in, there's a lot of virus. We need more help. Come and help us kill off the viruses. Those immune cells come in, they've produced some more signals to tell more immune cells to come in. At the same time, they're producing these cytokines, or these bombs, that are facilitating killing some of the the cells that are infected. And this ends up causing a lot of problems for the patient. And so in the context of cytokine storm in the lungs, you're going to end up having a lot of tissue damage within the lungs. And this tissue damage is going to prevent you from breathing. And that's why a lot of patients who are susceptible to cytokine storm end up needing to go on ventilators. And because the tissue damage is so extensive, they're not able to actually heal, and they end up succumbing to the actual immune response rather than the virus itself. And so to prevent cytokine storm, the best way that we can do this is by stopping the immune system or dampening the immune response. And the way we do this is by treating the patient with anti-inflammatory drugs such as dexamethasone. And there is a fine balance with this, because if you dampen the immune system altogether, then the virus is replicating. And so a lot of the time, doctors will not only give dexamethasone to dampen the immune system, but they'll also give the patient antivirals to also be killing the virus at the same time. Some of these can be pricey, and so that's why it's not typically a first thing that's done. I think now that, a year into COVID or the pandemic, we've been able to multiply a lot of these drugs now, but early on, we didn't have a whole lot of these. A lot of hospitals were running out. And so that's why we were getting a lot of patient death. So here is a nice diagram of the overall view of the immune system in response to COVID. So you have basically the virus getting into your lung airways. You have resident macrophages or macrophages that live within your lungs that basically clear, any pathogen that might be present, or, typical maintenance of, cells that naturally die and cleaning up the material. But in the context of infection, macrophages are going to alert your T cells to come and help us clear these viruses. And what ended up happening with these T cells is, like I was saying, the CD8 T cells are cytotoxic. So once they come in, they're going to end up killing your cells that are infected. And if there's a lot of cells that are infected, you're going to have a lot of cells that are going to be killed. And the body naturally will try to repair itself. But like I was saying before, when we breathe, we're breathing with our lungs that expand naturally. And there's specific types of lung cells that are able to facilitate that expanding. Now, if you end up having tissue damage within your lungs, what ends up happening during the repair system is you have a certain cell type called fibroblasts that basically will secrete certain molecules to kind of fill in where that cell had died. That filler that they're putting in isn't elastic like your typical cells that were killed. And so if your cells are being repaired, but they're not elastic anymore, then you're not going to be able to expand your lungs as well. And that's where a lot of people start to develop that shortness of breath. And when that ends up occurring, basically, patients should be seeing a doctor immediately so they can get put on antivirals and anti-inflammatories, because if not, it's going to end up becoming severe enough where more immune cells are going to be coming in and destroying everything, and you're going to end up on a ventilator. So here I have two prime examples of lung tissues from a healthy individual versus a COVID lung. And so typically, like I was saying, in a healthy lung, so all of this space is where air typically is going to be. And you have very thin cells that cover the alveolar space. And so these are going to be the cells that are able to expand. Now, when you have that tissue damage, like I was saying, your natural healing process is going to end up secreting, I think it's collagen, if I'm not mistaken, but secreting a lot of this filler into where there was some of that damage. And that filler is going to end up creating a lot of problems when it comes to being able to expand your lungs. And so you can see prime example, the difference between these two. You have a lung section that is basically not as open as a normal healthy lung. And this is what's happening to a lot of COVID patients. And this is why a lot of them develop shortness of breath. And this is why a lot of them end up succumbing to the infection. Here's a nice diagram of some of the stages of the disease. So basically most people, are going to end up experiencing flu-like symptoms or no symptoms at all. And then the immune system will clear it, and we're okay. And then there's about 15% of patients that develop that shortness of breath, but they're able to get their immune system under control and clear the infection. And a lot of the time, some of these patients end up staying long-term with some of the shortness of breath. And then this last stage, stage 3, where it's hyperinflammation, where you're having all of that whole cytokine storm that I was talking about. This is where a lot of patients start to die. And scientists and doctors still don't know why there's specific patients that end up succumbing to this. As I mentioned in, I think, the COVID replication lecture, a lot of the patients that do end up succumbing have that trifecta of, they're hypertensive, they are diabetic, and have cardiovascular disease. So their body is already filled with inflammation. And so adding a whole nother layer of inflammation, specifically to the lungs where you're breathing or you're needing oxygen to, fuel the body, this is going to end up creating a lot of damage. And this is where patients end up dying. Does the filler ever get replaced? No, it does not. So this is why, and it's the same thing when it comes to people that smoke. The smoke itself is causing lung damage or tissue damage. And so you're going to end up having immune cells that are going to try to fix the problem, but they end up putting filler in. And that's why, as time goes on, people that have been smoking for years, they're not able to breathe so much because of all this filler that is being put into their lungs, and they're not going to be able to expand as much. And there's a couple of different, forms of how the body responds to that, but we're not going to get into that. And that's all I have for you guys. I know it was a kind of a dense lecture, but I hope most of you guys were able to follow along. And if there's something you guys are struggling with, stay after. I have office hours today. I can stay on right now and help with clearing up. But I want you guys to really understand how your immune system works, because it is so important when it comes to understanding how we respond to infections and how our vaccines and other therapies are utilized to help fight off infections in the future. I put the sign-in sheet in the link, so go ahead and sign in. You guys have 10 minutes to do so. I also have extra slides for visuals if you guys want to look at that, because I know it's sometimes nicer to see different ways of the immune system responding. But yeah, that's it. Any questions?